Hey guys, we're going to be talking about the 3 studies that involve the biological approach.
But first, the assumptions. The first assumption is behavior, cognition, and emotion can be explained in terms of the working brain and its effects of genetics, hormones, and evolution. The second assumption is similarities and differences between people can be understood in terms of biological factors and their interactions with other factors. And now we're going to be talking about
the background of Aice et al. The first thing that you need to know about the background is what congenital adrenal hyperplasia is, which is when female fetuses are exposed to higher level of androgens in utero. Research shows that females with CAH prefer more masculine toys over feminine toys. Studying monkeys helps us separate the effects of nature versus nurture because they have the same sex hormones as us, but they are not able to be socialized. Play is a voluntary act of self-pleasure, and we can choose what to play and what to play with. Sex is determined by sex chromosomes, which are typically XX or XY. Socialization is learning how to behave in ways that are acceptable to society, which is how we learn about gender roles, like what toys to play with.
Aice believes toy preference is not caused by socialization, but by socialization abilities caused by hormone levels. There are 2 aims of Aice et al. The first aim is to test if sex differences in children's toy preferences are a result from biological factors rather than socialization.
The second aim was to investigate if male or female rhesus monkeys had similar toy preferences to human infants despite no socialization with gender-ruled toys.
Now for the sample of Aice et al. To begin with, they had 135 rhesus monkeys, 14 of which already had hormone testing done, so they were removed from the study. 39 were too young to tell their sex apart, so they were also removed from the study. This leaves 82 rhesus monkeys, 61 of which female, 21 male. Of these 82, 34 interacted the most with the toys. 23 female, 11 male. These were the only monkeys analyzed in the experiment.
Now we're going to be talking about the strengths. The first strength was animal ethics because they only broke numbers. The second strength was inter-observer reliability because there was a behavior checklist and 2 observers recording. The third strength is internal validity because there was multiple trials and counterbalancing and recordings. The last strength is that it was generalizable to all humans.
And now we're going to be talking about the weaknesses. The first weakness is internal validity due to observer bias. The second weakness is ecological validity because the monkeys were living in captivity. The third weakness is standardization because the monkey ripped apart a plush, so the trial was ended early.
Now for the results of Aice et al. Observers calculated average frequency and duration for each behavior by adding up the totals, then dividing them by each number of trials each monkey participated in. Then the total number and duration of interactions with each toy type was calculated. 73% of male monkeys preferred the wheeled toys, 9% preferred plush, and 18% had no preference at all. Male preferences were not affected by rank nor age. Females showed some preference for plush toys versus wheeled toys, but this difference was not significant. 30% of females preferred plush toys, 39% preferred wheeled toys, and 30% preferred no toy at all. Females who had no preference were ranked significantly lower in the social hierarchy than females who preferred plush toys. Females did not prefer the wheeled toys as much as the males, but they did prefer the plush toys more. There was a significant positive correlation between social rank and frequency of interactions with both types of toys. Now for the conclusions of Aice et al. Sex-typed toy preferences in humans may result from biological sex differences, as their observations show that rhesus monkeys have similar preferences to human infants without being socialized. Human toy preference, as with the monkeys, show hormonal influence on toy preferences.
And now, the men in Clayton.
The first you need to know about the nervous system. The nervous system sends electrical signals from the soma through an axon and out the axon terminals through synapses. Cells in the brain and the nervous system make up short-term communication in the brain and body. In 1953, Kleitman and another researcher named Asterinsky discovered REM. They discovered this on Asterinsky's 8-year-old son that those REM periods correlated with increased brain activity. Prior to this finding, it was always thought that the brain was in a restful state of sleep. Dement and Kleitman were interested in finding a reliable, valid way to measure when a person is dreaming!
And now for the aims of Dement and Kleitman.
Aim 1. The independent variable is whether the participant was woken up during REM or NREM sleep. The dependent variable is whether the participant can recall the content of their dreams. Yes or no? Do you remember your dream, yes or no?
Yes.
Do you remember your dream?
Yeah.
Okay. Aim 2. The independent variable is where the participant was woken up 5 or 15 minutes after the onset of REM. The dependent variable is the perception of whether they were woken up 5 or 15 minutes. Aim 3. The independent variable is the eye movement pattern. There's vertical, horizontal, mixed, and no pattern. However, the dependent variable is the description of their dreams. Aim 4. Correlation.
Covariable 1. This is the number of minutes spent in REM.
Covariable 2. This is the number of words in the corresponding dream narrative.
Now for the sample of the red climate.
There were 7 adult males and 2 adult females. 5 were used to study intensively, while the other 4 were used to confirm the results. The sampling technique, however, is unknown. But what we do know is that it took place at a sleep lab at the University of Chicago.
Go Chicago. Psychology investigated in the climate.
The encephalogram, known as the EEG, detects brainwaves through electrodes on the scalp. Four electrodes were placed near the eye and on the scalp. This measured eye movement and brain activity. Participants were then taken to a quiet, dark room to sleep. The EEG was ran throughout the night in the next room. Throughout the night, participants were woken at various times in both REM and NREM sleep by a doorbell that was loud enough to wake them. They were first asked whether they were dreaming or not, then to describe that dream into a recorder near the bed. The experimenter would sometimes enter the room to question the participants further about their dreams.
Now the results of demand excitement.
General results. All 9 participants had regular sleep patterns. This meant no sleeping or mental disorders, like insomnia.
REM never occurred immediately after the onset of sleep.
The average duration of REM was 20 minutes.
Now for the AIM results. AIM 1. Dream recall was the most in REM. 80% in REM and 7% in NREM.
Aim 2. 83% of participants correctly identified if they had been dreaming for 5 or 15 minutes.
Aim 3. Vertical eye movements included watching climbers on a cliff, climbing ladders, and throwing basketballs. The only report of horizontal eye movements was watching 2 people throw tomatoes at each other. Whoa! No pattern eye movement, including driving a car. Mixed eye movement included talking to a group of people, searching for something, or even fighting with someone.
Aim 4. Moderate positive correlation between the length of REM and number of words in a dream narrative.
And now the conclusions of the method clip.
Number 1. Dreaming occurs during periods of REM and episodes throughout the night. REM can be objectively measured by recording REM cycles in sleep. The strengths of demand climate. The first one is ethical guidelines. This is because they made sure to follow confidentiality. They didn't really give us much on our sample, so we don't really know much about the people. Next is standardization. They made sure that the participants slept in the same bed, the same lab room, and they all limited caffeine and alcohol. And now for the weaknesses of demand climate. The first one is generalizability. This makes sure that it could represent the population. There was only 9 people and 5 were males, so the results weren't that accurate. The last one is ecological validity. This is because they slept in a lab, so their sleeping conditions could vary on how comfortable they felt with all the wires attached to them.
And now, Ozell.
Psychology investigated in Hosel et al.
Greta Hosel was a psychologist, neuroscientist, MBSR, and yoga teacher. What is mindfulness? Mindfulness is a stress reduction technique that improves well-being. This includes gentle yoga, body scans, meditation, and more.
What is localization of function? This is the idea that specific brain structures are responsible for specific behaviors.
Hippocampus is a part of the brain that associates with memory and emotion. White matter transfers information. On the other hand, gray matter collects and processes information.
MBSR stands for mindfulness-based stress reduction.
MBSR is a technique combining meditation with a scientific approach. This is an 8-week-long program that includes 2.5-hour-long meetings. What is body scan? This is slowly becoming aware of sensations in each part of the body. What is mindful yoga? Gentle stretching, slow movements, and breathing with a focus on the moment-to-moment experience. What is sitting meditation? This is developing awareness of breathing, sensory information, and emotion. FFMQ stands for 5 Facets Mindfulness Questionnaire.
These are the methods of Hosel.
This is a field and lab experiment.
This is the independent variables. This is whether or not the participant did MBSR. This is independent measures. The data collected before and after the study is repeated measures. This is the dependent variables. Number 1. This is gray matter in certain brain structures and throughout the brain. Number 2. This is the FFMQ score. Number 3.
This is the time spent on mindfulness exercise in between weekly group meetings. This is a longitudinal design because it was taken over the course of 12 weeks. Hosel used opportunity sampling and they gathered people from 4 MBSR groups at Center of Mindfulness in New Hampshire. There was 33 participants, all of them were right-handed, all of them were healthy and they aged 25 to 55. All participants saw MBSR to limit stress and they all had limited meditation experience.
This is a participant doing yoga.
This is a participant doing meditation.
This is a participant doing a body scan. This is the control condition participant taking the MRI. This is the experimental group participant taking the MRI.
This is the experimental group participant taking the FFMQ. This is the controlled condition participant taking the FFMQ.
There were decreases in the PCC in the control group, but there were no changes in any structure in the experimental group.
The body scans, meditation, and yoga improved the left hippocampus. The meditation and body scans improved the PCC.
Now the conclusions of Hosel.
Structural changes in these key brain regions can occur as a result of participation in regular mindfulness practice even after just 8 weeks.
And now the strengths of Hosel.
One strength is ecological validity because this was their normal lives and normal activities.
Another strength was reliability. This is because they used the FFMQ.
And now the weaknesses of Hosel.
One weakness is ethics because it broke lack of deception and protection from harm. Another weakness is standardization because the participants got to choose whenever, wherever, and whatever.